Hello everyone, I hope you're all doing well. So today we're finally starting on part 5 of the textbook. So we're finally getting into this section on causal claims. And so we're going to start this section with chapter 10, which is an introduction to simple experiments. So we're going to go through a bunch of the different ways that you can set up or design your experiments and some of the pros and cons of each of these. So for our overview, we're going to start by talking about 2 examples of experiments because these examples will then help us illustrate some of the points we want to make throughout the chapter. We can talk about our experimental variables and some of the terms that go along with it, why we can actually use experiments to support causal claims, whereas we haven't really been able to do that before now. Talking about 2 different types of designs, our independent groups and our within groups. And then we'll get to the end and bring back some familiar topics by looking at those 4 validities again, but specifically looking at how we can use those while looking at causal claims. So that is our outline. So let's get started. So our first example is a study conducted by Mueller and Oppenheimer in 2014. And the people who did the experiment are less important than the details of the experiment itself when it comes to this class. And their experiment was set up to look at basically how students do using different types of note-taking methods. So they wanted to see which method of note-taking was the most effective. And so they had 67 college students recruited to come into a classroom. Usually they got students to come in pairs, not always, and they would prepare the classroom in advance. So sometimes the students would show up and it would have laptops set up ready for them, and other times the students would show up and the classroom would be set up with notebooks and pens. Now the students would be told to take notes using the method they were given, so either a laptop or a pen and paper, while watching 5 different TED Talks that had been selected by the researchers. So after the students watched the videos and took their notes, the researchers took the participants elsewhere to spend 30 minutes doing another activity, something to distract them and keep them from thinking about the lecture that they had just watched. After that distraction activity, the participants would be tested on what they had actually learned from that TED Talk. And so on the screen here, we have the results of what they found. And so when they were questioning students, when they were testing what they had learned, they asked 2 types of questions. They asked factual questions and they asked conceptual questions. And so factual questions would be sort of the what I've referred to in this course as definition type questions. Do you remember what this is, or what is this particular fact? Whereas a conceptual question is something that's testing your understanding of a topic. So they wanted to test both kinds of questions. And then the way that this graph here is set up is that we have a score that they received, but it's standardized. So the average on this standardized axis is 0. And so here, we have 2 different colored bars, pink being for laptop notes and blue being for longhand or written notes. And so both of the groups, even though we don't have error bars as I would like to see, both of the groups are very close to 0 for factual questions. So they performed pretty much average on factual questions. There's no clear winner in terms of which method of note-taking was better here. But if we hop over to our conceptual questions, once they started asking students about their understanding, then we see that people who took longhand notes, who had written notes, they performed much, much better than people who were using laptop notes. So this would be showing that people who took notes on the laptop scored lower than average, and the people who took notes on pen and paper scored higher than average. And so you can see the difference between the groups. So our researchers would then conclude that taking longhand notes makes you perform better on conceptual type questions. And because we're doing experiments, theoretically, they can make that kind of a claim, that taking notes a certain way causes you to do better at certain types of questions. Now, for our second example, we can look at the idea that the method of presentation of food will affect how much food that you eat. And so in this situation, we're looking at some researchers from Cornell University back in 2012 decided to set up an experiment to see if the serving bowl size, so the size of the container that food is served from, had an effect on portion size, so on how much food that students would take and eat. So the participants were randomly assigned to be in either the large bowl or the medium bowl condition. So then they would have these students come in to a kitchen during a lunch session, and the participants were asked to serve themselves from a communal bowl that was either large or medium sized. And so the students' plates were weighed before they ate pasta and then afterwards to account for how much food they had actually eaten. And so over here, we're going to look at graph A first, and this is just looking at which condition the students were in, what size serving bowl were they eating from, the medium or the large, and then how much pasta they served themselves. And so here we have that our medium bowl people served themselves less pasta than the large bowl students. And then this graph is kind of showing the same information but presented just a little bit different because they converted how many grams of pasta would be how many equivalent calories consumed. So you still see that the medium bowl people ate fewer calories than the people who served themselves from the large bowl. And so the researchers concluded that the size of the serving bowl would influence how much pasta people served themselves and how much they ate. All right, so now that we have those two examples out of the way, we're going to keep referring back to them to illustrate different terms and concepts and designs as we move through the chapter. So to get started, we're going to start with some terms. So when we're talking about an experiment, we've already kind of touched on what makes an experiment different from a correlational study, that makes it different from our early frequency claims, but we're going to put it on paper and talk about it very briefly one more time. And so here we're saying that an experiment is set up so that at least one variable is manipulated and at least one variable is measured. So we have some measured variables and some manipulated variables. So our simplest possible experiments would be one measured variable and one manipulated variable. Whereas I have conducted experiments where I've had a dozen manipulated and measured variables each. So they can get very, very complicated very, very quickly, but we're going to stick to the more simple side of things, at least for now. So when we're talking about a manipulated variable, this is the one where you can assign participants to a particular level of the variable. So from our note-taking example, our manipulated variable would be what kind of notes were the students taking. And the levels that we're talking about, the different conditions they could be in, are going to be computer or longhand. Did they take notes on the laptop or did they take notes on pen and paper? For our measured variable, This is basically where we're just recording what happens, usually in response to a thing that's been manipulated. So usually we're going to be recording some kind of behavior or attitude, looking at self-reports or behavioral observations or even physiological measures. So still with our note-taking example, the measured variable would be how they performed or how many questions they got correct when testing their knowledge. Now, you're not always going to see the terms measured and manipulated variables. What you are more often going to talk about are independent and dependent variables. So our let's start with independent because that's the way I've written them on the screen. So our independent variable, sometimes shortened to just IV, is our manipulated variable, the one that the researcher manipulates. So which note-taking method did they use? And so when I talk about conditions, conditions are also called levels, and these are the different types of independent variable conditions you can use. So one condition would be longhand notes, the other condition would be laptop notes. And the researcher selects the conditions or the levels that are used in a particular experiment. Now, we can also have our dependent variable, or DV, and this is our measured or outcome variable. And the way that I always remember the difference between these, I don't know if I've said this before, is that the dependent variable will change depending on our manipulation of the independent variable. So I always remember that the dependent variable depends on other factors. You might also see people talking about a control variable. And a control variable would be any variable that an experimenter holds constant. Now, if you remember back to when we first defined a variable, you might realize that a control variable is actually a misnomer, because variable means something that is changing, something that has multiple levels. But a control variable is something that is being held constant. So despite this term being poorly named, you might still see it. And if we want an example of a control variable in the study looking at pasta consumption, they could control the type of pasta that was used in the experiment. So both the medium bowl group and the large bowl group were served macaroni because they controlled that particular variable. So I guess if we're going to be talking about experiments, and we've spent all of this time building up to talk about experiments, and specifically the fact that we can use experiments to now start talking about causal claims, then maybe we should reevaluate our 3 criteria for causation, because we've been constantly pointing out how they don't work with other methods, with other types of studies and setups. But now we're finally into the realm where these 3 criteria can actually be applied and used. So by now you should know all of these fairly well. We have the first criteria is covariance. Is there a relationship between the cause variable and the effect variable? Are we seeing that there is actually a correlation here? We also need temporal precedence. Does the cause variable come before the effect variable? And internal validity is usually us asking about, are there any alternative explanations for the result that we're seeing? And so we can go through each of these. So experiments establish covariance. In the 2 example studies that we looked at, both of them showed covariance between their independent and dependent variables. Now, covariance is looking just a little bit differently in our experimental setups than what we were talking about with our correlation studies. But here, covariance is going to be indicated by a difference between group means. So are you seeing a difference in height between those two bars on a bar graph? So in our Posttest study, the participants in the large bowl group ate more than the participants in the medium bowl group. By having independent variables with multiple levels, we're getting multiple groups. So if you think back very early on in the course when we mentioned the importance of comparison groups, having a condition, something else to compare a particular experience to, we're bringing that back here. So our independent variables let us answer that question of compared to what? Because now we can compare between two different groups. And those two different groups have two different experiences because they're different levels of that particular independent variable. And so if we are going to talk about types of groups, so sometimes in an experiment, you're going to just look at two different levels of exposure. Maybe you want to look at people who take notes on computer and people who take notes by hand. Those are two different options. But sometimes you can see special cases, things like control groups. And a control group would be where you have a no treatment condition, which we can't really do when looking at our examples. So you can't evaluate how much pasta do people who don't eat pasta eat, because even trying to say that right sounds weird. But you also wouldn't necessarily, well, I guess, thinking about it. In our note-taking group, we could have a control group of people who just watched the TED Talks but didn't take any notes. So that would be one way to include a control group where they didn't have any treatment condition. And then all of our other groups where they do have a treatment condition, those would be called our treatment groups. So when there is no treatment done, or there is no exposure or no manipulating part there, that's the control group. And then once you have the extra levels brought in where there's some kind of treatment given, then you have a treatment group. And sometimes you have a special case where your control group could be a placebo group. So that's when your control group receives no treatment, but it's done in such a way to hide the fact that they're not getting treatment. So if we talk about a placebo, that would be something like a sugar pill that masks the fact that the people taking the pills aren't actually getting any kind of medical treatment. So having a placebo group lets you control for all of the other things going on, like maybe people's mental attitude. Maybe they don't like taking pills. And if your control group then just doesn't take pills, then there's something else going on that you have to account for. So our placebo groups are specialized control groups that let us control for even more factors, trying to do our best to keep everything consistent. All right, so our next of the three validities, we are in, make sure I say it right, our three criteria for causation. Everything has a name. So our second of the three criteria is establishing temporal precedence. And this one's actually pretty easy for experiments because you are controlling when people are exposed to things. So researchers make sure that the cause variable, the independent variable, the one that you are manipulating, comes first. Then we measure the effect variable, the dependent variable, and we see how it changed depending on that original manipulation. So in the Posttest study, the experimenters manipulated the independent variable, the serving bowl size, and they did that first because that's how the experiment was set up. So they put out a large or a medium bowl. And then after the students had interacted with that variable, then they measured the dependent variable to see how much food they had taken. So there's that temporal precedence where the independent variable comes first. Now, our last of our three criteria is that internal validity. And so this one's a little bit more complicated. So you can't necessarily say that all experiments establish internal validity, because internal validity is going to depend very strongly on how your experiment is set. So a good experiment, a well-designed experiment, can establish internal validity, but we still have the potential problem where a poorly designed experiment or an experiment where the researchers missed something could still lack internal validity. So when we're investigating internal validity, we have to be just a little bit more critical to figure out if this is in fact a well-designed experiment, and following if we can trust it to have internal validity. And so now when we've been talking about internal validity, especially when it comes to making causal claims, we pretty much just focus on ruling out potential third variable explanations. We want to be confident that the independent variable, the thing that we're manipulating, is what's actually causing the change in our dependent variable. We want to make sure there isn't something else going on. And so when we talk about this something else that's potentially going on, we can call those things confounds. So these confounds can confuse or threaten our internal validity. So an example in our PASTA study would be we could have had a confound if the different groups also received different kinds of pasta. So if our large bowl group also received more appetizing pasta, maybe it's a better kind of pasta than the medium bowl group, then that could cause a problem. That would be a confounding variable. If that was the case, we couldn't quite be sure if it was the bowl size or the pasta quality that was actually affecting how much pasta the participants were eating. Now we can call this a design confound, and we can actually care about this the most specifically when you're seeing this variability, that second variable, happening alongside the independent variable. So, like in my example, where the large bowl group also got better tasting pasta. That's when you see what's called systemic variability. So that second variable changes along with the independent variable. So that's when it's causing problems, when it's a real confound. But if you've ever thought hard about how research happens, there are all sorts of other things that are going on. So maybe on Tuesdays, you have Bob is your research assistant, and Bob makes pasta just a little bit better than everybody else. But if we're randomly choosing which group we're having on each day, so maybe sometimes Tuesday is a large pasta bowl, and maybe sometimes Tuesday is a medium pasta bowl. So because that better tasting pasta is happening sort of equally spread between the two groups, we're seeing what's called unsystematic variability. So the effects are randomly distributed or haphazardly experienced by both groups. So in those cases, if you can spread apart those effects, it's a lot less of a problem. So unsystematic variability is reasonable. Like you always want to try and avoid it as much as possible, but if you can't avoid it, you split it between the groups and it should cancel out between the two groups. Whereas if you have systematic variability, if Tuesdays are always the large pasta bowl and Bob always makes better pasta on Tuesdays, then you have a actual design flaw with your experiment, and it's something that you have to account for by either rerunning your experiment or finding a way to counterbalance that again. Another thing that we should be concerned about or looking for when thinking about internal validity is selection effects. And so selection effects are when participants in one level of our independent variable are systematically different than the participants in another level or levels of that independent variable. And so again, we run into this systematic differences. So it's causing a problem when there is a major difference between our different levels of the independent variable. And so to illustrate this, we can talk about a specific example. So back in 1987, researcher Lovis conducted a study specifically to test an intensive therapy for children with autism. So they wanted to have a more intense therapy to help children with autism. And so some students or some children with autism would receive this new and intensive therapy, whereas others would continue to receive the old treatment that was much less intensive. But these children weren't randomly assigned to the groups. Apparently, the researchers tried to randomly assign them to groups, but they had parents requesting to be switched between the different groups. So some of the families lived too far away and couldn't come into the city all the time for the more intensive treatment. So they couldn't physically make it into the city all the time to be doing the more intensive treatment. So by default, they got put in the less intensive, the old treatment group. And on the other side of it, some parents heard that there was a more intensive treatment available, and they asked to be switched from the less intensive treatment group to the more intensive treatment group. And so our random assignment has pretty much disappeared because people were self-selecting to be in the different groups. And so the researchers in the end found that the autistic symptoms of children in the intensive treatment group showed more improvement than those in the normal treatment group, but there was a selection effect that was influencing these results. So families in the intensive treatment group were actually probably systematically different from those in the normal treatment group, because these were the parents that were willing to drive long periods of time, they were willing to travel a lot, and they were willing to show up a lot more often for these treatments. So if these parents were willing to put in that much more effort, and not even just willing, but even able to, there could also be a socioeconomic status here, where if these parents were more wealthy, they could afford to take time off work or afford to have someone else go to these treatment sessions with their child. So there's this sort of dividing line between the people who could do the less intensive normal treatments and the people who were able to do the more intensive and more time-consuming treatments. And so the researchers ended up concluding that it's actually impossible to determine if the improvement came because of the treatments or because of the types of families that were receiving the different kinds of treatments. So another way to say it was, if your family has the time and the money to be able to go to these more intensive treatment sessions, they might also have the extra time and money to devote more effort into helping these children at home. So there are extra confounding variables here that you can't really control for in this situation. And so to get around this, researchers could enforce random assignment. They could say, no one's allowed to switch. We have to just use random assignment. But for this study, it's difficult because What if the parents physically can't make it to the more intensive treatments and then they drop out of the study? So that's something you have to keep in mind and that you have to watch for to investigate internal validity, because there are other factors that can be going on. And these are things that the researchers themselves should be aware of, but also you as a critical-minded reader, you should also be aware of those as well. And so we've talked about random assignment to try and get rid of those selection effects. But there's another option that you can also use, and this is called matched groups or matching. So instead of just randomly assigning people into group 1 or group 2, into intensive treatment versus normal treatment, you could also try and control for some extra things. So in cases where random assignment doesn't necessarily work, you might be able to use this matched sampling method. And so you could pick a variable, an important variable, that could vary between the groups that you want to control for. So, for example, maybe you want to look at IQ or high GPA, which might be correlated with IQ. And so what you would do is pick all of the students that have very, very high IQs or high GPAs. So maybe people who have 4.0 GPAs. And what you would do is then randomly assign those people with the very high GPAs to be in either group 1 or group 2. And by doing that, you ensure that there's an equal split of high GPA students in each of the groups. Then you'd go down to the next highest level. And you'd say, okay, so for people who didn't quite get a 4.0, maybe let's look at people that have a 3.5 or higher. And then you can randomly assign them into group 1 or group 2. And you continue to systematically go through your participants and split them evenly between the groups. So you're still using random assignment, but you're also taking that extra step to try and control for this potentially confounding variable of GPA. So sometimes this is an effective alternative to pure random assignment. So our next section for this chapter is where we're going to look at two different type of group setups for your experimental designs. So we're going to look at independent groups and within groups designs. And these all have multiple different names that you might hear them referred to as. So our independent groups design, you might also hear it called a between subjects design or a between groups design. And this is the one that I feel like people are most comfortable with or most used to in terms of experimental designs, because this is where you have different groups of participants that are placed at different levels of the independent variable. So you have one group that is shown the large pasta bowl and some groups that are shown the small pasta bowl. So, or some participants are shown the large and some are shown the medium pasta bowls. And those two groups are made up of separate individuals. So they are independent groups. There's no overlap between those groups. And we're going to talk about two types of independent group designs. We're going to look specifically at post-test only and then pre-test, post-test designs. But before we get into that, I also want to talk about the alternative, which are our within group designs. And these are also sometimes called within subject designs. And these are different than the independent group designs because each participant is presented with all levels of the independent variable. So our first one, the independent groups design, is when you have different people in different groups, and those groups each get exposed to one level of your independent variable. In a within groups design, you really only have one group of participants, and everyone gets exposed to all levels of your independent variable. So with our pasta example, maybe you get participants to come in twice. And the first time you show them the large pasta bowl. And then the second time they come in, they have the medium pasta bowl. And you want to see how they change their response each time. And when we talk about the within groups designs, we're going to talk about concurrent measures and repeated measures. But we're going to focus on the independent groups first, and then we will come back to our within groups. So first up, we have our post-test-only designs. And in these setups, participants are randomly assigned to our independent variable groups and are tested on the dependent variable just once. So this is what we've seen much so far. So you have random assignment, you get put into group 1 or group 2, and you are given a particular treatment. We can use our laptop versus longhand notes example. So you randomly assign them, you get them to watch the video and take notes on the laptop, or they take notes on their piece of paper, and then you test the dependent variable, their performance on a test at the end. So there is only a single testing going on here. And so this can work for a bunch of different setups. But you're measuring that dependent variable only once and only after exposure. Now, if we look at the other option, our pre-test post-test design, you might now be able to figure out that you are going to test before and after. So here, participants are randomly assigned to their independent variable groups, same as before, but here they're tested on that dependent variable twice before and after they are exposed to that manipulation. So we can look at an example here where they wanted to look at the effects of mindfulness training on scores on your GRE. So the idea being that if you are more mindful and calm, that you would perform better on these particular tests. So people could be randomly assigned to this mindfulness class, or they could be randomly assigned into a control class that has nothing to do with test scores, so a nutrition class. And so before they attend those classes, they wanted to get a baseline for the participant scores on verbal GREs. So they tested both groups at the beginning of the experiment to see how they score on these particular types of questions. Then they sent both groups to their classes, one for the mindfulness class, the other group went to a nutrition class that was unrelated, and then after attending these classes, they retested them to see their performance on these verbal GRE scores. And so if we look at the graph over here, our data is going to be presented just a little bit differently than everything that we've seen so far in this chapter. So we have our score of accuracy on the verbal GRE, so how well did the student do? And then across the bottom, we have our two groups, the group that went to a nutrition class and the group that went to a mindfulness class. But this time we have 2 bars per each, one that is our pre-test, their measure beforehand, and one that is the post-test, their measure afterward. And so here we would set up our statistics to ask the specific question of, did the group improve on their scores between the before and the after? So did going to a mindfulness class help them perform better on this task? So our nutrition group has the 2 bars very, very close together. And this time, they finally have error bars included. So error bars showing basically the spread of our data, and these two have their error bars overlapping quite a bit. So this tells us that there is no difference in the before and after tests for the group that went to a nutrition class. So as we assumed, going to a nutrition class didn't help them improve on these scores. But for our mindfulness group, their before test is less, and it's significantly less because we see this little asterisk here, and we have this no overlap between our error bars that helps visualize that significant difference, where they performed better after attending that mindfulness class. And we can also look at the fact that before they went to these classes, both of our groups were pretty much the same. Their score before any training, before being exposed to different levels of our independent variable, were very, very similar. And in fact, the bars are almost identical in height. So we are getting kind of double information here, showing that the groups were the same before the treatment and that there's a difference in only one of the groups after treatment. So this pretest post-test design actually ends up giving us even more information. So when we ask the question, which design is better, your gut feeling might be, well, it has to be the pretest post-test because it's giving us that extra information. It also gives us that opportunity to confirm that the two groups were the same before you expose them to any kind of manipulation. But the real answer is that it's going to depend. Our pretest post-test design isn't always the best. So the situation that we're in, the specific kind of research that we're doing, determines which design is better. So in some situations, it's not really possible to use a pretest post-test design. So, for example, if we were using our pasta example from the very beginning, and you wanted to test how much pasta people can eat, it doesn't really make sense to test how much pasta they eat before you expose them to a manipulation to test how much pasta they can eat, because they'd be full and they wouldn't be able to eat any more pasta. So sometimes it's not really viable to be measuring a particular dependent variable beforehand. So it's really difficult to set that up sometimes. There are limitations on what you're measuring and how you measure it. So not always viable. But in other cases, you can come up with a way to make a pretest post-test design work, and it can help you actually get that initial establishing confirmation that your two groups are the same before treatment, which is really, really helpful. But we should keep in mind that that doesn't mean that our post-test designs are bad. If you have a well set up post-test design, it can still be very powerful. So if you have random assignment and you're manipulating the variable, then you can still be fairly confident in your internal validity. So you don't necessarily need to confirm that random assignment worked if you've done it well. So which is better is it depends on the situation and what makes sense. All right, so we're going to move on now from our independent groups designs, and now we're going to start looking at within group designs. So we'll start with repeated measures design, and this is actually one that I have used in my own research quite a few times. So a repeated measures design has participants measured on the dependent variable more than once. Basically, you're going to measure them on that dependent variable each time they're exposed to a level of the independent variable. So if you remember our within groups design, each participant gets exposed to all of the levels of our independent variable. And so we want to measure their response to each level of the independent variable by measuring their response on the dependent variable every single time. And so we can have another example to illustrate this kind of a setup. So we can look at an experiment by Boothby and colleagues back in 2014, where they used a repeated measures design to look at whether shared experiences intensified that experience. So the idea here being, does having another person present make an experience more intense? And so they had 23 college women came into the lab to do a study, and each participant was joined by a female confederate. And if we remember, confederates are basically research assistants. They're people who are in on the experiment. Basically, they've been told by the researchers to behave a particular way. So they will act like they're another participant, but in fact, they're there as an and so in this experiment, they had a participant and a confederate sit side by side, facing forward, and they weren't allowed to speak to each other. So there was no interaction between them, but they were both there together. And so they had chocolates to sample. That was the experience that they were going to be sharing, was tasting chocolates. And the participants were told that the two chocolates that they would be sampling were different. But, of course, it's an experiment, so the two chocolates were exactly the same. The only thing that was differing was how the participant was experiencing the chocolate. So in the first exposure, they would taste the chocolate while the confederate was in the room. So two people sitting together would both taste the chocolate, and then they would rate how much they enjoyed the chocolate. Then they had the confederate leave the room. They got people to rate everything on their own. So taste the chocolate alone, and then rate the chocolate again. And so remember, this is the exact same chocolate. The only thing that's differing is whether or not there's someone else in the room with you also having the chocolate. And their results showed that people in the shared experience rated their liking of the chocolate higher than when they were alone. So they enjoyed the chocolate more, they enjoyed that experience more when there was a person there to share the experience with. Now, this is a repeated measures design because we have them exposed to one level of our independent variable, tasting the chocolate with someone present, and then they are measuring that dependent variable, their enjoyment of the experience, immediately following that exposure. Then they have the next exposure, so the next level of our independent variable, tasting that chocolate alone, and then they are rating that experience. We have that dependent variable being measured again, following the second level. So we are repeatedly measuring our dependent variable after each exposure. Next, we're going to talk about concurrent measures design. And I want to start right off the bat by realizing that I made a typo and I forgot to change the definition here. So this should in fact be concurrent measures design. I just didn't change it from the last slide. So make sure you update your notes with that. But in a concurrent measures design, participants are going to be exposed to all levels of our independent variable at roughly the same time. So instead of like with the repeated measures design, where you experience one level and then you're tested, and then you experience the next level and then you're tested, for a concurrent measures design, you're exposed to multiple levels at once, and there's going to be a single dependent variable measured. And usually it's going to be preference. So they're usually getting at preference here. So an example would be, they do these kinds of studies fairly often with infants, and they use gazing as a measure of preference. So you can imagine they have an infant and they'll hold up two pictures, a female face and a male face. And they can measure which one the infant prefers by measuring which one they look at for longer. So if the infant looks at female faces for longer, then our dependent variable would record that female face is their preference here. So you can expose them to multiple things, ask them to choose, and that choice is our measured dependent variable. And because they're picking from multiple independent variables, female face or male face, they're getting exposed to them all at once. So that's how we have our differences between repeated and concurrent measure designs. All right, so if we want to look at the advantages of using these within group designs as opposed to using our independent group designs, we have three major points that we can look at. So the first here is that for our within group designs, the participants in our groups are equivalent because they're the exact same participants. So when we're doing our independent groups designs, you're sort of hoping that your two groups match. Maybe you're using random selection, maybe you're using a matched samples design, but you're trying to make your groups equivalent. For a within groups design, you don't really have to try to make your groups equivalent because they're the exact same people. They are equivalent. And when you have a setup like this, those individuals get to act as their own controls. So the only difference between the two situations, between being exposed to one independent variable and being exposed to the other level of the independent variable, you can attribute it to those levels of the independent variable. It's not because the people are different. It's not because there's something else going on. It's going to be because of that change in exposure, for the most part. The second point that's good about within groups designs is that these designs give researchers more power to notice the difference between conditions. So by having the same people in those two different groups, we're getting rid of sort of extra stuff that's going on. So when you have different people in different groups, there's lots of extra noise going on. And we called that unsystematic variability. We were looking at it earlier. So maybe if we were looking at that pasta study, maybe some people are gluten intolerant and aren't going to eat a lot of pasta. Maybe some people love pasta and are going to eat a lot no matter what group they're in. And by having those individual differences and having that variability, that reduces our power, that reduces the strength of the statistics that we can run. And so when we're talking about power in this context of stats, this is the ability of a study to show a statistically significant result when the IV actually has an effect on a DV. So if you have high power, it means that you're going to be able to find an effect. You're going to find a difference between the groups if there is a difference between those groups. If you have a poor setup or if there's something else going on, you can end up with low power, which means that even if there is a difference between the groups, your data isn't going to let you see it. So obviously, having more power is a good thing. You really want to have as much power as possible, and these within group designs allow you to do that because it's reducing all of that noise, all of that extra variability between the groups to give you a clearer picture of what you're looking at. And then our third point is that within group designs actually require fewer participants overall than other designs. So imagine that you want to run an experiment and you want to have 20 participants in each condition. So if you're doing a independent groups design, you'd need 40 people to fill two groups because each group would have 20 people. And we can actually look at this over here. So you'd have 20 people in condition 1 and 20 people in condition 2, and they're different people, so your independent designs will have 40 people total. But if you're running a within groups design, the 20 people in condition 1 or group 1 are the same 20 people as in condition 2. So you actually end up using half as many people, which is really, really nice. Especially if you wanted to set up something that had, say, multiple conditions, more than 2. Maybe you're looking at 5 or 6, then the difference in size becomes massive. So this is another benefit here. All right, and here we have another oops on my part. This is a duplicate slide that we already talked about, and I'm not quite sure why it's here. So in your notes, again, just cross this one out. I'm not quite sure why that's there. So next, we want to start talking about our three criteria for causality, specifically as they apply to our within group designs. So we've already kind of talked about them in terms of our independent group designs, but now we're going to focus on the aspects that are specific to within group designs that can potentially mess with especially our internal validity. So quick reminder yet again of our three criteria. That's covariance, temporal precedence, and internal validity. So with covariance, as long as we're manipulating our independent variable and we have these different comparison conditions, if we find a statistically significant result that's showing a relationship, then we have evidence for covariance. That one's still pretty easy to evaluate. For temporal precedence, same thing. If we're manipulating and controlling the order that participants are exposed to things, then we know that the independent variable comes before the dependent variable because that's how we're setting it up. It's when we get into internal validity that's, a little bit more complicated. So we're asking again, are there potential third variable explanations? And for within groups designs, one of the biggest problems we can run into are order effects. And these can be a potential fairly large problem for internal validity. Now, an order effect happens when exposure to one level of the independent variable influences the responses to the next level of that independent variable. So basically, is being exposed to one level also affecting how you respond to the next level? Does the order that you are exposed to these particular levels affecting your behavior? And there are two major types of order effects that we can talk about. The first is the practice effect. And this occurs when participants either get better at a task because they're practicing at it, or they get worse at a task because they're tired from it. That's sometimes also called the fatigue effect. So if you imagine we're testing how well people perform at a puzzle game. How quickly can they solve this puzzle? The more they're doing that puzzle, potentially the better they're getting at it. So their improvement in performing this particular task might actually be due to the fact that this is their second or third or fourth time completing the task, and it might not have anything to do with what treatment group they're in or what condition they're being exposed to. And then the same thing with fatigue. Maybe the thing that you're asking them to do is energetically draining or even mentally draining. So if you're asking them to solve a complex problem, maybe they do okay the first time, but as they get tired, they're going to be worse at solving that. Not because of whatever condition they're being exposed to right now, but because they've already done this and they're tired. So obviously, having these extra things involved confuses our results. It makes it difficult to pick apart what's actually causing the change in performance. The next type of order effect is a carryover effect. And this happens when some kind of contamination from one condition carries over to the next condition. So this is a huge problem for things like drug studies. But we can use an example of maybe you want to look at the difference between how people perform on a test when they have coffee that has caffeine in it versus decaffeinated coffee. So if the people are in a group where they get regular coffee first, then they're going to have caffeine in their system. And caffeine persists in the system for hours. So you would have to wait days to be able to test them again. So you would have to watch what the participants are given and how long that persists in their system to be able to make sure that you're not seeing a carryover effect and that any behavioral differences are actually from your manipulations. So if these order effects are such large problems, then what the heck can we do to get around them? And the first of these is to use what's called counterbalancing. And counterbalancing is kind of actually really simple. All you do is present the different levels of your independent variable to the participants in different orders. So for our coffee study, maybe some people get the caffeinated coffee first and some get decaffeinated coffee first, but you make sure that you wait a day in between testing or something like that. There are lots of different ways you can counterbalance. One of the easiest, at least on small scale, is to use full counterbalancing. So if you have two conditions, there are two orders. So if we have A and then B, so this would be condition A and this is condition B, some people will get A first and then you test their dependent variable. You would measure the dependent variable. Then they get exposed to condition B and then you would measure the dependent variable. Again, the other group of people would get B first and then A. And the best way to set this up is that you would divide your participants, you would divide that pool of participants into two, and then half of them would be assigned to get A first and then B, and half would get assigned to be exposed to B first and then A. So they are divided into groups, but they're still getting exposed to the exact same thing, just in different orders. The thing to keep in mind with this is the more conditions that you have, the more levels of your independent variable, the more complicated this becomes. So A then B or B then A is simple enough, but if you have three conditions, so A, B, and then C, so you can have A, B, C, you can have C then B then A, you can have A then C then B, so on and so forth. And I believe for 3 different conditions, you end up having 6 possible orders. And then if you move up to 4 different conditions, you end up with, I believe it's either 14 or 16 different orders. So this increases exponentially very, very quickly. So full counterbalancing isn't always a viable option. So, okay, what do you do then if full counterbalancing isn't an option? Then you can use partial counterbalancing. And so partial counterbalancing is great when you have lots and lots of possible orders of exposure. So partial counterbalancing, as you might expect, would be when you're only exposing them to a subset of the possible condition orders. So an example of this would be maybe you would present your 100 stimuli in randomized order. You could set up a computer to randomly select stimuli from a pool of 100. So you don't have enough hundreds of participants for everyone to get all of the unique possible orders, but you can use randomization to make sure that you have a bunch of different orders. You could also use kind of a systematic way of having partial exposure, and that's called a Latin square. And I actually really enjoy Latin squares. So these are a systematic way to ensure that each condition appears in each position at least once. Meaning that, so here we have 4 possible conditions, A, B, C, and D, and you have A at the beginning at least once, you have B at the beginning at least once, C at the beginning at least once, so on and so forth. So you can set up this sort of a setup. This is called a Latin square, and it's just a way of making sure that you have A in the first position once, A in the second position once, third and fourth all once. So this isn't all of the possible ways that you could present for different exposures, but it does allow us to make sure that each condition appears in each position at least one time. So those are how within groups design works and some of the advantages of it. Maybe now we should also consider some of the disadvantages of these within group designs. And when we're considering these, there are 3 major disadvantages for these within group designs. So the first is the fact that, especially repeated measures designs, they can have order effects, and those can be very problematic for your experiment. But we've already proposed a solution for that with counterbalancing. So it's a potential problem, but we have a way around it as long as we're thinking about things properly. The second disadvantage is that sometimes it's not practical or possible to set up an experiment in this method. So for example, if you wanted to look at different methods to teach children to ride a bike, and so method A is one way to teach them to ride a bike, and method B is a different way to teach them to ride a bike. You can't teach them to ride a bike more than once. Once a child has learned to ride a bike, they know how to ride a bike. So you can't really use this method for that. So once that condition has happened, once that teaching method has occurred, your participants are permanently changed. So if that exposure permanently changes your participants, it doesn't really make sense to use a within groups design. Our third problem is that sometimes exposing your participants to all levels of that independent variable, it can change the way your participants act. And so this changing the way participants act is called demand characteristics or experimental demand. And basically what we're saying is that if your participant sees every different level of your independent variable, they might pick up on some cues that lead them to guess what the experiment is testing. And people will adjust their behavior to be in line with what they think you're looking to find. So with our pasta experiment, if you have them in on consecutive days, and the first day there's a large bowl of pasta, and the second day there's a smaller bowl of pasta, they might make the connection and say, oh, they're probably seeing how much we eat based on presentation. So maybe I'll eat less on the smaller bowl day to be in line with what they're looking for. And, some people will go the other way and they'll intentionally mess with your experiment. But either way, having the participants figure out what's going on and adjust their behavior defeats the purpose of setting up an experiment to understand their natural behavior. So all of these are things to watch for. Now, the next slide actually covers something really interesting. And this is a comparison between our pre-test post-test design, which was one of our independent groups designs, and a repeated measures design. And The reason that we want to compare these two is because on the surface, they seem very, very similar. So in this case, if we look at sort of the basic setup here, our pretest, post-test design at the top, and then repeated measures at the bottom, both of them are testing or measuring that dependent variable multiple times. And so they kind of look fairly similar here. But we can point out some very critical differences. In our pretest, post-test design, it's an independent groups design. So the participants are only ever exposed to one level. So here, exposed to level A or exposed to level B. So they're never exposed to both. Whereas for our repeated measures design, by their setup, you have to be exposed to both. So they get exposed to level A and level B, though some people will experience it in a different order, B then A. So it's all about what they're exposed to and when, not just the fact that they're being tested or measured on that dependent variable multiple times. So just a quick pointing out the fact that though they seem on the surface quite similar, they are both very different. And so, of course, coming to the end of the chapter, we're going to want to start looking at these causal claims critically. And the way the textbook does that is to interrogate our claims with our 4 validities, our construct validity, external validity, statistical validity, and internal validity. So we're going to cover each of these. By this point, we've done this so many times, and there's so much similarity between different techniques that we're going to spend a lot less time on these than we have in the past. So let's start with our construct validity. And so we're asking, how well were the variables manipulated and measured? And so this one is a little bit different than what we're used to because we're not just caring about how well they were measured. Now we're introducing the fact that some of our variables are manipulated. So dependent variables, that's old hat. That's stuff we've talked about before, where we can ask about how well that dependent variable or variables were measured. So if we think of our note-taking example, when they were testing students using factual questions versus conceptual questions, were those questions actually testing factual knowledge and conceptual knowledge? That would be thinking about the construct validity of those variables. But for our independent variables, these are the ones that are being manipulated. So now we want to ask, how well were they manipulated? So you want to know, did they actually do a good job of adjusting or manipulating these particular levels? So this is kind of related to operationalizing the thing that we're looking at. And again, in our note-taking example, it was a fairly straightforward manipulation. People were given a pen and paper, or they were given a laptop. And so you could quickly evaluate how well they manipulated those independent variables by seeing if people on the laptop took typed notes and if people with pen and paper took written notes. It's as simple as that. But it's not always that easy. So sometimes you want to be a little bit more sure than what we've seen with that example. So you can do a couple of things. The first is a manipulation check. So researchers can add in an extra dependent variable that will give us an idea that the experimental manipulation actually works. So we don't really need this for our laptop versus paper notes thing, like I said, but let's say that we were looking at an experiment that was manipulating how people felt. So maybe we wanted to see how people's behavior changed when they were angry versus when they weren't angry. A manipulation check could introduce a questionnaire that asked people how upset they were, something that measures their level of anger, because you'd want to make sure that the people who were in a group that was supposed to be angry were actually angry. but you'd also want to check that the group that isn't supposed to be angry isn't angry. So introducing an extra step, an extra dependent variable, something that you can measure that would tell you, have they been manipulated the way that you wanted them to be manipulated? And that can help you be a little bit more confident in the construct validity of that independent variable. Another way that you can do this is by using something called a pilot study. And a pilot study is usually a simple, fairly stripped down experiment that's using a separate group of participants from your real study that you would complete either before or, in rare cases, sometimes you'll do it afterwards, to figure out if the data that comes out of that is what you're looking for. So instead of a separate variable introduced to the experiment that you're working on, you could run a separate experiment that just looks at how effective is this method of making people angry. So instead of introducing a couple of questionnaire questions, maybe you would have an experiment that's just, does scolding people for their behavior make them feel angry or not? So your pilot study would help you understand the construct validity of the method of manipulation. And so, best case scenario, you would want to do this first to make sure that the method that you want to use is going to work. You can get around this if other experiments have used this method before and they've already done all of the checks to make sure that it's an effective method of manipulation, but not always. Sometimes you do a pilot study after the fact because your data is confusing. So this is more of a not best practices, but sometimes what you observe in real life. And that is, you might run a study and come to the end of it and be like, wait a second, why isn't there a difference between groups? There really should be a difference between groups. And so as you try and eliminate other potential problems, other potential confounds, you might go back and run a quick pilot study to make sure that your method of manipulation actually worked, to eliminate that as a possible problem with your results. Not necessarily the best order to do things in, but sometimes that's what happens. So good research doesn't necessarily need to use these, but it can, depending again on how things are set. Oh, I did not mean to scroll there. All right. Another thing to keep in mind for this construct validity is to actually look at your theory testing methods. What is it that you are trying to test? So in our laptop versus longhand note-taking experiment, the researchers originally thought that laptop note-taking would be a disadvantage because students would be taking verbatim notes, meaning that they would be copying down what the instructor is saying, or what, I guess, the TED Talk is saying word for word, instead of thinking about it and processing it and putting it in their own words. And so if that's what their theory is based on, then they could actually measure, okay, well, how many verbatim notes were taken? Were the people on laptops taking more word for word notes than people who were handwriting? And if you're curious, that's exactly what they found in that study. So by measuring specific things that were directly related to their theory, they can give themselves just a little bit more construct validity in that situation. All right, so next we can look at external validity. So this is when we're asking who or how or what can we generalize our results to. And so again, we've already done quite a bit of this. So for generalizing to other people, mostly we want to be looking at how the participants for this particular experiment were selected. So was it a random sample? Are these individuals representative of the population that they were drawn from? And is the population that they were drawn from the same as the population that you want to generalize to? That's a really important one, especially where a lot of the studies we talked about today were conducted on university students. So they wouldn't necessarily be able to directly generalize those results to the general population without jumping through a few extra hoops. When we're talking about generalizing to other situations, so with our note-taking study, we looked at taking notes while listening to taped TED Talks. Well, could we apply that to real-life lectures? Could we make the same assumptions about live lectures if you were listening to someone lecture actually in the classroom with you? And for this case, you kind of, again, have to critically think about it and maybe run another study. So by replicating your results, by having another experiment that's very similar to yours, but applying to another situation, or heck, even applying to other groups of people, by having that agreement between multiple studies, by having your results replicated, you might be more confident in your ability to generalize those results to other people and other situations. Now, the last point to consider here is, what if your external validity is poor? So we've talked about the fact that in a lot of experiments, people don't necessarily care too much about external validity. They might sacrifice external validity in order to have good internal validity. So they might want to have better experimental control. So you can end up in situations where, yeah, you don't have fantastic external validity. You've only run your experiment on undergraduate university students who are specifically in a laboratory setting. So this one study might not be something that you can apply to a lot of real-world scenarios. So again, you really want to look at other research. You want to make connections with other studies, see if these results can be replicated in other scenarios in order to be able to generalize. So sometimes you have to accept that your external validity is poor and that you really can't apply anything more broadly. And that's okay, as long as you're aware of the limitations. So next we can talk about statistical validity. So how well do the data support the causal claim? Does our data actually fit what we were investigating? And again, this is going to be a lot of similarity to what we spent lots of time talking about for correlational research. So the two big things you care about are, one, was there a statistically significant difference? Was there a difference between the groups? And two, how large is the effect? So you have to keep in mind that having a statistically significant difference is not the same as having a large effect size. You can have a statistically significant difference, but it could be a difference between very small effects. If you have a really well-designed experiment with lots of subjects in that experiment, lots of participants, you might be able to point out a very small or weak effect that doesn't really mean much in the real world. So you have to kind of evaluate the difference between statistical significance and what that actually means in a practical sense. So usually a larger effect size is going to be something that we care more about because it's a stronger relationship. And then the one major difference that we want to consider is that in experiments, instead of using R, when we're comparing differences between groups, we're mostly going to be using D, or Cohen's D, which is going to behave very, very similar to R. And actually at the bottom here, we have Cohen's guidelines for effect size strength, where we've already seen this side of things, where an R of 0.1 is small or weak, an R of 0.3 is medium or moderate, and so on. But we can also look at D in terms of small or weak, medium or moderate, and large or strong. And these are scaled up just a little bit because they're not the exact same as R's. So here, a D of 0.2 is small or weak, 0.5 is medium or moderate, and 0.8 is large or strong. So same idea, a little bit of a different application. And then last but not least, we can talk about our internal validity, which is something that we've focused on pretty much throughout this entire lecture. So we're going to summarize it down into 3 important questions that you should ask about internal validity. So the first thing we want to ask is, were there any design conflicts? So is there some other variable that accidentally co-varies or systematically varies along with your intentional independent variable? So with our pasta group, they made sure that everybody had the same kind of pasta. In the lecture notes video, they made sure that both groups saw the same videos. So have you controlled for as many things as possible? Are you sure that there aren't any design conflicts? Second, if you used an independent groups design, did researchers control for selection effects? Did they use random assignment or matching to make sure that there wasn't something else against? 2, if an independent groups design was used, did the researchers actually control for selection effects? So did they use random assignment or matching to make sure that there wasn't any systematic variability in the participants that were assigned to each of the 2 groups? And then for our third point, if you used a within groups design, did the researchers control for order effects by using counterbalance? Or is order effect not an issue because of some other considerations to take into effect? So just a bunch of ways that you can think critically in terms of our internal validity. And we're going to talk more about this and some of the extra details about experiments in the next couple of chapters, where we go into sort of less simplistic experiments and get into some of the nitty-gritty on those. So I will see you guys there.